Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami microbiologist. Today in this video we are going to see butyrate disk test. In the previous videos lecture we talked about 18 biochemical test. If you remember, if you don't know about the biochemical test and all the biochemical tests which we already talked, so you can go back to my channel, or I will give the link in the description box. Then you will find out the biochemical test. There will be a biochemical test playlist. Then you will see all the biochemical tests one by one video. Okay, now it's time to talk about the butyrate disk test. So let's start the video. First of all, we have butyrate disk test. We also call it BTT in short form. So it is actually the test in which we mostly find Moraxella species, or especially Moraxella catarrhalis. This is the bacteria, and we mostly find out this type of the bacteria. Now what's going on in this type of bacteria? They utilize a molecule which we call that bromochloroindole butyrate, or this is actually the molecule that is present in a medium and mostly utilized by the Moraxella species. Now what's going, this bacteria is actually causing a type of different types of infections, like we have otitis, sinusitis, conjunctivitis, bronchitis and pneumonia. These are the infection that is caused. Now we have large number of M. catarrhalis strains that producing an enzyme which we call that beta lactamase enzyme, and due to the production of this enzyme, what will happen, this bacteria will get resistant to penicillin and ampicillin antibiotic. Now this test, if we not perform, so we have conventional test method that require 24 to 48 hours. So that's why if we compare this conventional test with this butyrate disk test, so what will going, it will be very rapid, easy to perform, and best test for the identification of Moraxella catarrhalis bacteria. Now what happened in the laboratory, we have the enzyme that is produced by this Moraxella, and that enzyme is called as butyrate esterase. Now once this enzyme is produced, so what will happen, the hydrolysis of the substrate, like we have bromochloroindole butyrate, and with this molecule will be break down, the hydrolysis of this substrate will be break down, and it is possible by this butyrate esterase enzyme. Then we have the principle of this test. So it's very simple. So we have bromochloroindole butyrate that is acting as a substrate. So it is what these are the substrate. Now what's going in this case? In this case we have an enzyme which we called it esterase that is produced by Moraxella. Now this esterase will hydrolyze this bromochloroindole substrate. Now once it is hydrolyzed, so what we get endo-oxide due to the presence of oxygen. Now when we get this, so due to the presence of oxygen we get indigo. Now what is indigo? Indigo is the color molecule, we call that chromogenic molecule, that is appear blue to blue violet in color. Okay, now after this what we get, we have to examine this hydrolysis of methylomebrophyryl substrate, which actually producing fluorescent under the UV light, and we mostly detect it under the UV light, and due to this way we get the fluorescence of this color. So that's how we can detect the blue to blue violet color in this case. Now if we see the tested microorganisms, so we mostly tested those microorganisms that are gram-negative diplococci, oxidase positive, and mostly those bacteria that are growing on blood agar and showing white colonies. So these type of the bacteria should be tested. We have different types of the requirement. We have personal protective equipments, we have other laboratory material that we used for the other experiment, general laboratory material, but the most important requirement are disk impregnate with bromochloroindole butyrate. Okay, this is actually the a kind of liquid material, so we have to uh, dip our disk. We have sterile wooden applicator stick, or we have bacteriological loop, we also call it wire loop. We have distal water, petri dishes, slide or tube, long way, UV light, etc. So these are the material that we need. Now let's see the procedure. First of all, what we have to do, we will take the disk. This is the disk, white disk we have. Okay, we will take it from the vial and we'll place it into the clean glass slide, or we can also say into a petri dishes. Now after that, we will put one drop of the distal water or deionized water onto the disk, and due to this way, then we will take heavy bacterial inoculum. This is really important, heavy bacterial inoculum. Now once we take heavy bacterial inoculum, then we will mix it, or we will scrap it onto this this antibiotic disk, onto this disk. After 5-minute incubation at room temperature, we will observe the color. So remember, this is the positive color, as you can see under the UV light. If you observe, this is the positive color. Okay, and this is the negative color, as you can see. This is the negative result. This is also the bacterial colony, this is also the bacterial colony. In this case there is no change, but in this case the changes occur, violet color changes. So this is the positive result. We have the uses of this test. We mostly use this test to identify the M. catarrhalis that is differentiated from the N. gonorrhea. So mostly Neisseria gonorrhea and M. catarrhalis, these two bacteria are what? These two bacteria are the positive, oxidase positive, and gram negative diplococci. That's why there is a big differences between them. So to differentiate these two bacteria, we perform this Butyrate test. Now the objective says that we mostly use this test to detect the M. catarrhalis from clinical samples that hydrolyze butyrate substrate by the production of butyrate esterase. Let's see the limitation of this test, what type of the error we arises while performing that test. So remember, if we provide longer incubation, so what will happen, it will give false positive result. That's really important. So within 5 minutes the result will be observed. Next is that sometimes we get false negative result due to the small inoculum. So we have to take heavy inoculum to get a positive correct positive result. Next point says that the test we are going to do, it should be performed with the addition of the other tests, like we have carbohydrates, fermentation, oxidase reaction, gram staining, morphology. So all these tests are also performed for the complete identification. Now we have some other type of the Moraxella species, like we have Echinella acinetobacter. These two types of the are the Moraxella species, and it will give positive or weakly positive reaction. So you have to be very careful. We have some other bacteria, like we have Staphylococcus, Pseudomonas. It will also give the positive result. Okay, so that's why other biochemical tests are really necessary to confirm the Moraxella bacteria 100%. So in the next video we will explain salt tolerance test. I hope this video is helpful for you. Thank you so much for watching.